Hello guys, welcome to another video. In this video we're going to learn about the gene cloning, and in this session we are going to cover about these questions such as what does the term the cloning mean, what is gene cloning, how does it differ from cloning an entire organism, why is gene cloning done, how is gene cloning accomplished, what are some of the ethical consideration regarding gene cloning. So let's get started. What is the term cloning mean? Cloning is the process of producing genetically identical individuals of an organism, either naturally or artificially. In nature, many organisms produce clones through asexual reproduction. Cloning in biotechnology refers to the process of creating clones of organisms or copies of cells or DNA fragments. Then, what is gene cloning? To clone a gene is to make many copies of it. Act of making many identical copies of gene can be an exact copy of natural gene. Gene can be an altered version of natural gene. Here you can see gene, chromosome, gene 1, gene 2, and this is gene. And making copy of gene is known as gene cloning. Here you can see in the picture whole organisms are cloned to but differently. Here you can see that cloning is occur from one section to another, different types of various plants, and here you can see cloning is done. Then why we clone DNA? A particular gene can be isolated and its nucleotide sequence determined. Control sequences of DNA can be identified and analyzed. Protein, enzyme, RNA function can be investigated. Mutations can be identified. Gene defects related to specific diseases. Organisms can be engineered for specific purposes, insulin production, insect resistance, etc. Then, how is DNA cloned? We can clone DNA in two types: cell-based DNA cloning and cell-free DNA cloning, which is PCR. Here you can see cell-based DNA cloning. DNA is extracted here from blood sample and DNA. Restriction enzymes such as Lac-Operon cut the DNA into small pieces. Different DNA pieces cut with the same enzyme can join or recombine. Then here you can see DNA cloning. Lac-Operon restriction enzyme cut the DNA, then sticky end and non-sticky end, sticky end joins together, and then recombinant DNA are created. The action of restriction enzymes Lac-Operon gives a sticky end. Then DNA cloning to bacterial plasmids, small circular DNA additional to a bacterial regular DNA are cut with the same restriction enzyme. Here you can see plasmid and a gene for antibiotic resistance, which is cut with the same restriction enzyme. A chunk of DNA can thus be inserted into the plasmid DNA to form a recombinant. Here you can see the foreign DNA which is inserted into the plasmid, and we can create a foreign recombinant DNA. Here we can see the mismatch desired recombinant plasmid. Then DNA cloning 3. The recombinant plasmids are then mixed with bacteria which have been treated to make them component or capable of taking in the plasmids. Here we can see this insertion is called transformation. Here you can see recombinant plasmid and E. coli host are inserted into transformed cell. Then DNA cloning 4. The plasmids have naturally occurring genes for antibiotic resistance. Bacteria containing plasmids with these genes will grow on a medium containing the antibiotic. The others die, so only transformed bacteria survive. Here you can see transformed cell and non-transformed cell. This is nutrient medium and ampicillin. Then overnight growth and ampicillin resistance colonies are formed here. So this is DNA cloning 4. Then antibiotic resistance. The medium in this petri dish contains the antibiotic kanamycin. The bacteria on the right contain kanamycin, a plasmid that is resistant to kanamycin, while the on the left has no resistance. Here you can see left has no resistance and right is resistance. And note the difference in growth. Here you can note difference. There is no resistance and there is resistance. DNA cloning 5. The transfer bacterial cells from colonies on the medium. Each cell in a given colony has the same plasmid and same DNA. Cells in different colonies have different plasmids and different fragments. Here you can see restriction on large DNA molecules. Then we can cut the gene with Lac-Operon restriction enzyme. Then we can insert into the vector, and the resulting recombinant DNA molecules are produced. All will produces colonies. Here you can see the colony are produced. So this is all about the DNA cloning. That's all for today. We will see you in the next video. Thanks for watching.